So the Metagenomics Center is one-stop shopping basically for anybody wanting to do microbiome research. We provide all the full range of services for anybody looking to characterize or understand polymicrobial communities. So that includes DNA extraction, library preparation, sequencing, and all the informatics you would need to do. So there's a lot of new evidence coming out looking at the role of the gut microbiome on human health. Obviously this is a big factor when we talk about things like inflammatory bowel disease and colorectal cancer, but there's a lot of new evidence indicating that the gut microbiome can influence disease susceptibility to things like autism spectrum disorder, cardiovascular disease, other non-intuitive types of disease processes. We're very interested in how the gut microbiota can affect animal models of those diseases, and so we're very interested in refining animal models to make them more reproducible, generate more reproducible data, but also to make sure that they're really translatable to the human condition. We have an intimate knowledge of animal models, just based on our expertise and experience working in comparative medicine, working with the mouse and the rat centers. We really have a really good understanding of what to expect, what's normal in a mouse or a rat in a research setting.
The Mutant Mouse Resource and Research Center, which is a mouthful, so we often refer to it as the MMRRC or the Mouse Center, is actually a consortium of 4 centers that are funded by the National Institutes of Health, the NIH. And what our role is to serve as a repository for mutant mice. And when I say mutant mice, those are mice that have been genetically engineered to either have a genetic deficiency and or an addition of a gene and or combinations of different genetic variations or mutations. One of our very important issues is to aid in reproducibility of the models that we have. We can maintain these as genetically pure and genetically identical so that that's not an issue. So we have created 8 colonies now of mice that are basically the same genetically, but that harbor different intestinal microbiotas, and these represent the intestinal or the gut microbiotas that are present in the biomedical community. The overall functions are to recruit and procure mutant mice that have been produced by scientists throughout the world, to cryopreserve their germplasm so that we have them in perpetuity and we will never lose those models. Great expense went into making them. To distribute those models to any other scientist that needs or wants them. And then the 4th thing we actually do is to perform research that benefits, is very broad-ranging research that benefits the mouse community as a whole and the functions we have as a repository.
Sure, the National Swine Resource and Research Center was established in 2003 to serve as a core facility for National Institutes of Health funded investigators.
So the goal of the Swine Center from its beginning has several aspects. One is cryopreservation of models from others so that they're not lost. We also make brand new models to distribute to researchers to meet those needs. And we've begun to provide more and more services as biomedical researchers realize that they need some assistance when they begin working with pigs. So pigs are becoming more and more popular near the end of research for translational medicine. We would argue they need to be at the beginning too. The fruit fly and C. elegans, a worm, and the mouse have been very important over the years, but they don't represent mammals in total. And pigs are just a much more appropriate model for physiology and size to humans.
So when people decide what model they should use, first of all, you have to consider availability. And mice and rats are readily available. Pigs aren't as readily available, but they're more readily available than a primate. You have to consider the phenotype. An example here is if we try to make a model of cystic fibrosis, the pig actually recapitulates all the symptoms of cystic fibrosis that humans get. Mice and rats don't do it. The size counts. Again, if you're developing devices and trying to come up with treatments and therapies, you need human-sized dosages, you need human-sized devices. Pigs are much more similar in size.
So the Rat Resource and Research Center, or the RRRC as we call it for short, is a unique NIH-funded resource that was put into place to archive and distribute important rat models. One of the things that we've recognized is that the microbiome, so the bacteria that are part of an organism, are really important aspects of disease or normal health. And so we are interested in looking at that connection between the microbiome and the rat models. Just like we pay attention to the genetics of our models and we pay attention to their disease phenotype, like what characteristics they have, do they get disease, do they not? We're encouraging people to also look at the microbiome as another aspect of models that you have to pay attention to, because if you don't, it could affect research reproducibility. And I think partnering with the MUMC now allows investigators to have one more avenue to really characterize their rat models, either the ones they receive from us or ones they're developing in their own lab. And so it's kind of, they can come to one place. It's like one-stop shopping for anything that has to do with rats, and that includes being able to characterize the microbiota.
My role here is understanding the role of the microbiome and its relationship to genetic disease. So we study animal models of genetic disease. And so studying the mouse and the rat, there's lots of variation in phenotypes. And so to understand those phenotypes, we needed to really understand the microbiome. The MUMC really allowed me to do that. So we can use the next generation sequencing technology that's now available to really characterize the microbiome. And then we use the MMRRC, the mouse center, and the rat center here to rederive our animals with different microbiomes. And being able to control both the microbiome and actually understand what it is has allowed us to see the effects on our primary phenotypes. And so that's had a really profound effect on how we do research here. Here at the University of Missouri, we've got the mouse center, the rat center, and the swine center, as well as the capacity to do the sequencing both of the host genome and of the metagenome. And then the ability to really transfer those microbiomes between species and understand their role in understanding genetic disease. So I think this is really crucial, and I think it's an amazing place to be. And the resources available through the MUMC make this really all available to everybody.
We really like thinking outside of the box, and so we welcome collaborators to come to us with new ideas and things that might not be possible in their mind, and try to figure out a way to make it work.